여기 와주신 여러분께 정말 감사드리고요. 그리고 또 초청해주신 브릭과 이제 한비서 여러분들께도 정말 감사드립니다. 제 이름은 신지은이고요. 여기서는 그레이신으로 활동하고 있습니다. 소개시켜 주신 대로 저는 이제 콜롬비아 대학에서 이제 연구원으로 활동 중이고요. 콜롬비아 대학 중에서도 이제 Zuckerman Institute이라고 이제 Mind Brain Behavior라는 이제 팀을 가지고 이제 브레인 사이언스를 연구하는 인스티튜트이고요. 또 이쪽 콜롬비아에서는 톰슨 패밀리 파운데이션 이니셔티브라는 이니셔티브에서 스콜러로 활동 중에 있습니다. 제가 먼저 이제 연구를 소개시켜드리기 전에 혹시 여기에 이제 또 콜롬비아 대학에 관심이 있어서 오신 분들도 있을 것 같아서 제가 어디서 근무하는지 잠시 먼저 소개드리고 이제 연구 발표를 시작하려고 합니다. 많은 분들이 아시는 대로 저희 콜롬비아 대학은 맨하탄 북부에 위치하고 있고요. 메디컬 캠퍼스가 가장 북쪽에 있고, 그리고 맨하탄 빌 캠퍼스, 최근에 생긴 신생 캠퍼스가 있고요. 그리고 모닝사이드 캠퍼스가 이제 각각 이제 세 곳에 이렇게 위치해 있습니다. 가장 관심 많으실 것 같은 메디컬 캠퍼스는 아실지도 모르겠지만 이제 굉장히 큰 규모이고요. 이제 의료 쪽 교수님들과 이제 베이직 사이언스 교수님들이 400분이 넘을 정도로 굉장히 큰 규모입니다. 그래서 정신도 굉장히 없고 이제 굉장히 활발한 연구가 진행되고 있는 곳이고요. 그리고 아까 말씀드린 대로 맨하탄 빌 캠퍼스는 이제 새로 생긴 이제 캠퍼스인데 제가 여기서 근무하고 있고요. 여기서 보이시는 이제 이 건물이 이제 뉴로사이언스 인스티튜트입니다. 에릭 칸달 교수님과 이제 또 리처드 엑셀 교수님 그리고 루이 코스터 교수님 세 분이 이제 디렉터를 같이 이제 맡고 계시고요. 보통은 이제 서킷이나 이제 베이직 뉴로사이언스 연구가 이루어지는데 이제 교류 연구들이 굉장히 활발하게 이루어지고 있는 캠퍼스이기 때문에 이제 메디컬 캠퍼스에 있는 분들과 굉장히 같이 협력 연구를 많이 합니다. 저 또한 이곳에 있으면서 제 대부분의 콜라보레이터들이 메디컬 캠퍼스에 있습니다. 그러면 이제 제가 이제 일하는 곳 소개를 마치고 이제 연구를 영어로 발표해 드리겠습니다. We often think of pain as a negative sensation that needs to be reduced and abolished. But at the same time, it is an essential function for our survival from potential injury and infection. The overarching goal of my research is to understand how our sensation of pain or nociception can be maintained properly. Pain treatment options are severely limited, and managing symptoms through painkillers, anticonvulsants, and antidepressants are some of the best available options. What if we can solve the pain problems for individuals suffering from pain? Given that prevalence of the chronic pain is extremely high, and in cases of US, over 20% of adult populations in the US, this would have a tremendous effect on individuals, families, and our society. Traditionally, sensory afferents that innervate the skin layer are the primary cell type that detects painful stimuli. But exciting evidence from recent studies indicates that keratinocytes in the skin and the immune cells surrounding these nerve terminals work together with neurons to detect and modulate nociception. These studies highlight that the understanding the nociception in their local context is critical, but how they crosstalk and change their relationship in pathological conditions are poorly understood. Therefore, my research aims to identify interactions between neurons and these non-neuronal cells that drive sensory dysfunction and the development of the chronic neuropathic pain. In today's talk, I'll focus on sharing our results from neuron ECM epidermis interactions. Model organisms like the Drosophila larva system depicted here can provide important insights into understanding the relationship between neurons and their physical substrates. The Drosophila larva epidermis is simpler, but has key characteristics conserved in the mammalian epidermal layer, so they can be used as a productive model to understand sensory neuron maintenance in its local context. These nociceptive terminals use the extracellular matrix and the epidermal cells as their physical substrates to grow on and connect to. It is tug-of-war between the epidermal cells and the extracellular matrix that determine where the nociceptive terminals will connect to. So the right balance between these three determines the integrity and the function of the nociceptive neuron terminals. So you can imagine that the too much force from the epidermal cells or too much force from the extracellular matrix could result in changing their branching pattern, structural integrity, and the disruption in the sensory function. I used Drosophila larvae as a primary model to study nociceptive mechanisms, and it is one of the most well-characterized systems to study the function of sensory neurons in vivo. Shown here in green are nociceptive neurons that are focus of my research. This and other types of somatosensory neurons are covering the entire body wall, as you can see from these two larvae. There are some anatomical differences from vertebrates, but like vertebrate sensory afferents, these terminals are nerve endings without a typical synaptic structure. This system also allows studying the sensory neurons in their complex 3D local environment, which is very hard to achieve by using in vitro system. In addition, their transparent body wall allows us to monitor changes in morphology and even changes inside the neuron in intact animals, such as protein trafficking, as you can see from this video from my current study. With a deep understanding of circuits and function of sensory neurons with different modalities, Drosophila peripheral nervous system serves as a useful model that identified many novel mechanisms relating to pain, neurodegeneration, and neuroprotection that later showed conservation in vertebrate animal models and humans. So in my study, I combined this Drosophila in vivo model and adult mouse in vitro and in vivo models to identify, investigate, and validate pathological mechanisms of peripheral sensory neuropathy. So peripheral sensory neuropathy is the focus of my research, which is a common yet debilitating condition affecting more than 20 million people in the US alone. It is a complex disorder without prevention or effective treatment options. Symptoms include pain, numbness, tingling in the hands or feet, which can greatly affect the quality of patient's life. A cardinal feature of peripheral sensory neuropathy is a loss of sensory afferents in the epidermis. These small fibers, pointed out by the arrow on the top panel, are the primary receptors for sensing pain. In healthy adults, these nerve fibers are frequently found in the epidermis, whereas in patients with peripheral neuropathy, these terminals tend to degenerate and sometimes completely disappear from the epidermis, as you can see from this example. Degeneration of these nerve fibers in skin correlates with disease severity, but it is poorly understood how these fibers change to result in major decrease in density. In some patients, these fibers become thinner and tend to overbranch, with an increase in varicosities before they start degenerating. These fibers also cluster in an unevenly distributed manner, as you can see from this example from patient sample. So I believe that understanding the cellular changes in neurons and substrates that precede the degeneration, such as this, is key to protecting these neurons at pre-pathological stages of peripheral neuropathy, and this is what I aim to answer in my research. So we have so far studied cellular mechanisms of how to commonly use chemotherapeutic agents. Bortezomib and paclitaxel can damage sensory neuron terminals in Drosophila and mouse models of chemotherapy-induced peripheral neuropathy. But today's talk will be focusing on paclitaxel studies. In today's talk, I'll refer to chemotherapy-induced peripheral neuropathy as CIPN. So paclitaxel, the focus of my study that I'll be presenting today, which is more commonly known as Taxol, is a common chemotherapeutic drug that is frequently used for treating solid cancers such as ovarian, breast, and lung cancers. It can kill cancer cells by stabilizing microtubules to interfere with cell division, resulting in mitotic arrest and apoptosis of the cancer cells. but at the same time, it damages sensory neurons and sensory neuron terminals and induces peripheral neuropathy in up to 80% patients in the long term. How paclitaxel damages sensory neuron terminals is under active investigation, but pathological mechanisms are still unclear, and we still don't have effective preventive measures or treatments in the clinic. So today, I'll first introduce my CIPM model and my quantitative methods to confidently characterize complex phenotypes. And I'll talk about cell surface receptor integrins, which we identified as a receptor that promotes healthy maintenance of nociceptive terminals, branch pattern, and function. This will be followed by characterization of the intracellular mechanisms of integrin-mediated protection, highlighting the importance of neuron to substrate interaction in healthy maintenance of nociceptive neuron integrity and function. In our CIPN study, we started with the hypothesis that dysregulation of cargo deliveries, which are required for the local connections and survival of the neurons, could cause the degeneration of arbors. Sensory neurons are one of the longest neurons in our body, as many of you know, and this inevitably puts their different cellular compartments into vastly different environments: proximal axon in the spinal cord, and peripheral axon along the body wall, and the axon terminals in the skin. As I showed you in the skin biopsy section examples in my earlier slide, peripheral terminals selectively degenerate from skin layers in many cases of CIPN. How does that happen? We think that as a dynamic structure within a constantly changing environment composed of the skin cells, these sensory afferents terminals require constant turnover of the cell surface protein, which are regulated by neurons' recycling and degradation pathways. So from here, we predict that disruption or change in cargo delivery precede the degeneration of the neuron terminals. So with this hypothesis, we predict that we investigated in our study that how paclitaxel leads to the cellular changes that eventually lead to degeneration, and whether we could prevent this change by facilitating the healthy maintenance of these terminals. So in our study, we first optimized the CIPM model using Drosophila by feeding paclitaxel to larvae in different doses, ranging from 1 to 30 micromolar. I'd like to note that this is not a really high concentration that you might see, but this is actually referring to the concentrations of paclitaxel that we use to bake up the food, rather than the actual concentration that the nociceptive neurons are encountering. In our experiments, we used the nociceptive neuron specific marker PPK-CD4-TDGFP in our experiments that allowed the observation of high order branches in both live and fixed animals. So using this marker, we found that the paclitaxel treatment to Drosophila larvae induced both pre-degenerative and degenerative changes in nociceptive sensory terminals. Similar to what has been observed from human patients, we found overbranching of thinner branches and increased varicosities and fragmentations from paclitaxel-treated animals. We found another striking difference that we observed was a change in their density of the density of the neurons. By utilizing an automatic quantitative approach, we first measured the local density of arbors across the entire receptive field, which was then assigned in the color map, as you can see from here. So from here, we've done several quantitations, and we found that the more nerve terminals are clumped in paclitaxel treated animals, while leaving other areas without nociceptive coverage. In the areas showing increased density in paclitaxel treated animals, we found that these arbors frequently overlap with one another as marked by red arrowheads. This was actually really interesting to us because it was reminiscent of phenotypes from mutants where neuron to substrate interaction is perturbed. As you can see from this control example, these neurons normally avoid overlapping of sister branches from same neuron through the process called self-avoidance. Self-avoidance is an important step in the organization of neurons shown in multiple systems, not only Drosophila sensory neurons, but also in vertebrate system. For example, starburst amacrine cells in the visual system, as well as the Purkinje cells in the cerebellum, they showed non-overlapping branch patterns, as you can see from these examples. This self-avoidance process ensures functionally appropriate coverage in the input and output field in our nervous system. And this is achieved by largely combination of two mechanisms. First, the contact-mediated repulsion between the branches, and second, the selective adhesion of branches to the extracellular matrix. We are interested in the second mechanism of self-avoidance, adhesion between sensory neuron branches and the extracellular matrix. This process is actually mediated by the cell surface receptor called integrins, which form heterodimers between two subunits alpha and beta, and bind to the ligands in the extracellular matrix, which then mediate bidirectional signaling between neuron and the extracellular environment. And as you can see from this image, from a previous study in Wes Gruber's lab, knocking out the integrins mimic the overlapping arbor phenotype that we found from paclitaxel-treated cells. This previous study also showed that integrin knockout decreased the maintenance of arbors and demonstrated that these phenotypes are actually due to a change in the interaction between neurons and the ECM. Also, in the literature, the integrin expression levels in different neuronal populations appear to correlate with a neuron's ability to regenerate. So we were encouraged by these studies, and we next investigated whether overexpressing integrins can protect nociceptive neurons from paclitaxel toxicity. So to test this hypothesis, we used the binary driver GAL4 UAS system in Drosophila that allowed nociceptive neuron-specific overexpression of integrins in our experiments. The yeast protein GAL4 binds to an upstream activating sequence UAS domain to activate the transcription of downstream sequences. So in our case, under the control of the nociceptive neuron-specific PPK GAL4 driver, we overexpressed both alpha and beta subunits of integrins in nociceptive neurons. Using this tool, we found that compared to their genetic control that you see on the left, integrin overexpressing neurons have significantly decreased number of overlapping arbors, indicated by these red arrowheads, as well as the degeneration phenotype indicated by these yellow asterisks in these images. We quantified these phenotypes that are shown here, magenta representing the data from wild-type animals treated with paclitaxel, compared to cyan, representing integrin overexpressing animals treated with paclitaxel. In both categories, integrin overexpressing cells showed significantly reduced overlapping arbors, and the varicosities and fragmentations together indicating that integrin overexpression protected the integrity and branch pattern of nociceptive neurons from paclitaxel toxicity. Next, we wanted to ultimately show whether the morphological changes in the cells and the prevention of these changes by integrin overexpression may be functionally relevant. So we asked whether the pain-related behavior of the larvae may have been affected by paclitaxel and how integrin overexpression may change this. Drosophila larva that you see here has a stereotyped escape behavior called nociceptive behavior against noxious stimuli in the forms of thermal, mechanical, or UV that are detected by polymotor nociceptors that I discussed in previous slides. In this experiment, as I'm showing you as still images to begin with, we used a hot plate that was heated to 40 degrees Celsius. In control, the larva rapidly responds to the heat and rolls to escape from the heat and then transition to crawl in a faster speed. In sharp contrast, we found that the paclitaxel-treated animals showed reduction in this nociceptive response, which is in the form of like often failing to initiate rolling or have a much shorter rolling sequence. So to first understand the change in the nociceptive function by paclitaxel in our model, we chronically treated the animals with different concentrations of the paclitaxel and matching vehicle concentrations. So from here, we found that the animals showed reduction in the nociceptive responses at 10 and 20 micromolar, which is resembling the loss of pain sensation or hypoalgesic condition shown in the clinic. We were encouraged by this result, but because these animals were chronically treated, we wanted to examine whether the damage in the nociceptive terminals were indeed driving phenotype before we move on to the next set of experiments. So to test this idea, we dermogenetically activated the second order interneuron called DMB, which is receiving the majority of the input from nociceptive neurons. This allows us to bypass the potential damage in the primary receptor and to see any potential effect from the effect of the paclitaxel on the rest of the sensory motor circuit. In our experiment, we found no difference between control and the paclitaxel-treated animals, which supported our hypothesis that the damage in nociceptive terminals is the major driver of this phenotype. So with this confidence, we next tested animals that are overexpressing integrins in nociceptive neurons. In integrin overexpressing animals, we found enhanced nociceptive behavior, which is indicating the functional protection of nociceptive neurons. This shows, together with our quantification, in addition to the morphological rescue of the neuronal integrity, nociceptive neuron-specific overexpression of integrins also protected the function of this neuron against paclitaxel toxicity. So after this, as we are continuing to investigate the changes in nociceptive neurons in the larva system, our morphological and functional protection results provided a strong premise to validate our results in a vertebrate model. So we move on to the mouse DRG sensory neuron experiments. And for the integrin augmentation, we targeted ITGB1, which is a major beta subunit in DRG neurons that makes heterodimers with various alpha subunits that you see here. And because augmentation of a single subunit can recruit other subunits to increase signaling in integrin pathways in the literature, we hypothesized that increasing integrin beta subunit alone prior to paclitaxel treatment would be sufficient to increase integrins to counteract paclitaxel-mediated toxicity. So we tested this hypothesis in DRG neurons, DRG sensory neurons, isolated from adult mouse in vitro. Briefly, we infected DRG neurons once they were stabilized in culture at 5 days in vitro, and then waited another week to start paclitaxel treatment so that we could see the effect of the paclitaxel and integrin protection in mature neurons. So here we have images from our experiment showing control cells transduced with the empty vector on the top panel, or a major integrin subunit in sensory neurons, ITGB1, in the bottom panel. We found that the transduction of the integrins alone did not cause any discernible changes in their morphological integrity or any differences in our quantification. But when we treated with paclitaxel for 72 hours, we found that there is a marked increase in fragmentations. In contrast, these were noticeably decreased when integrins were transduced prior to paclitaxel treatment. We quantified these levels of the degeneration by calculating degeneration index, and this corroborated our observation showing that integrin augmentation protected the DRG neurons from paclitaxel toxicity, as first demonstrated in our Drosophila CIPM model. So once we identified integrin as a target, we next asked what are the underlying changes in cells that could be prevented by integrin overexpression. So we thought that one of the ways that the cell surface proteins maintaining their levels is through intracellular control of the endocytic pathway. Endocytosed cell surface proteins are sorted in these early endosomes before they traffic to recycling or the degradation compartments that will determine the fate of the proteins. Integrins, as the receptors, are also using these endocytic pathways, and they maintain their levels preferentially by recycling rather than going through degradation and de novo synthesis. Therefore, we asked whether these endocytic pathways were perturbed, leading to the depletion of the total integrin levels, or even selectively leading to the reduced availability on the cell surface. So from here, I hypothesized that any changes in endolysosome motility or size may indicate a change in these endocytic pathways because similar changes were reported in other models of neurological disorders. And we further predicted that these changes may be linked to changes in integrin cellular turnover in nociceptive neurons. So to begin to characterize potential intracellular changes in nociceptive neurons by paclitaxel, we used two markers, REV4, which labels early and recycling endosomes, and SPINSTER for lysosomal degradation pathways. Due to time constraint, I'll be focusing on showing you REV4 results today, but SPINSTER results can be found from my paper. In our study, we monitored the motility of the moving vesicles by imaging a portion of the nociceptive neuron branches for up to 5 minutes at 1 or 2 frames per second. So in subsequent slides, you'll see a type of the stacked image called kymograph. And as you'll see in this cartoon, kymograph is a 2D graphical representation of the movement of these vesicles over time, with each row reflecting an individual time point. So using this type of the recording, we first monitored REV4 motility in nociceptive neurons in larvae that had been chronically treated with paclitaxel or vehicle control. Here we have a kymograph that shows the spatial position of REV4 in this ROI, with time shown here in y-axis. And as you can see from this kymograph, REV4 bidirectionally travels, ranging at different speeds in control animals. But in contrast, we found that the paclitaxel treatment largely abolished moving REV4 vesicles. So we used the semi-automated method to analyze these changes, and here is one of the quantification that we have done that showed significant increase in the proportion of the stationary REV4 in nociceptive neurons in paclitaxel-treated animals. And the full range of the quantified data can be found in our paper. So from here, we predict that the change in motility of the REV4 potentially leads to a change in the recycling route of many cell surface receptors, including integrins. So then, what about integrins? Do they really change from perturbation of REV4 mediated trafficking changes? So in order to answer this question, we directly assessed REV4 positive vesicles carrying integrins in nociceptive neurons. Here we have a 3D render image showing the integrins and REV4 that were detected based on their structure. To quantify these intracellular compartments with higher confidence, what we did was to image these cells using super-resolution microscopy and a machine learning-based quantitative approach using Elastic. We found that the REV4, we detected the REV4 and integrin positive structures in the nociceptive neurons using this way, and segmented according to the neuronal compartment for our 3D quantification. So here are maximum intensity projection of the cells that we used in our 3D analysis that I described in the previous slide. Integrin positive puncta are shown here in magenta, and REV4 positive puncta shown in green, and co-label structure appear as white. In control neurons, we found abundant and strong colocalization of REV4 and integrins, while colocalized vesicles were significantly reduced in nociceptive neurons from paclitaxel-treated animals. Our quantification of the density of the co-localized structure showed a reduction in all compartments, whereas the strongest reduction was shown from the dendrites, which are the nociceptive arbors in the periphery. So altogether, these results indicate that a reduction in RAV4 trafficking may likely reduce the availability of integrins in nociceptive terminals. So now we know that the recycling vesicles carrying integrins reduce after paclitaxel treatment. But this doesn't mean that the perturbation to recycling pathways actually led to the degeneration and dysfunction of the nociceptive neurons. So we next investigated when this trafficking chain starts for RAV4 positive recycling endosomes. And importantly, as you may remember from my earlier slide, we were interested in finding out whether this precedes morphological degeneration of nociceptive terminals. So instead of treating larvae starting from first instar, we treated larvae for either 48 or 72 hours with paclitaxel and monitored the nociceptive neuron morphology in live animals. So as you can see from here, both genetic controls and animals expressing RAV4 markers in nociceptive neurons, we found almost no change in the morphology by 48 hours. But we found strikingly more significant increase in varicosities by 72 hours of paclitaxel treatment. And the quantification is shown here. So based on the degeneration phenotypes, we determined 48-hour time point as a pre-degenerative stage and assessed whether we could detect any RAV4 trafficking changes. We found that the RAV4 in live animals showed a significant changes in both availability and the motility at this time point. As you can see from this quantification and this chymograph, the number of the RAV4 endosomes is reduced, and the stationary proportion of the RAV4 significantly increased by this stage. This supports our hypothesis that the change in the RAV4 mediated recycling precedes morphological degeneration of nociceptive neurons in vivo, and that this is not the byproduct of degenerating neurons. Our results also show that integrin overexpression in nociceptive neurons protected RAV4 trafficking following paclitaxel treatment. This points to an interesting hypothesis that the integrins may protect nociceptive neurons through both cell intrinsic and extrinsic mechanisms. Similarly, we also asked whether these cellular mechanisms are conserved in the mouse CIPM model. We used the same condition that was used in our integrin protection experiments in adult mouse primary culture, but this time we treated them with paclitaxel for shorter periods, 24 hours and 48 hours. Similar to our Drosophila results, we identified 24 hours as a time point before morphological degeneration. At this time point, we observed a significant change in the RAV4 endosome motility in adult mouse sensory neuron cultures, indicating that the trafficking changes preceded the neurodegeneration as shown by Drosophila experiments. So now, as a final result, I'd like to show you how we attempted to identify conserved integrin changes in recycling and further demonstrate changes of integrins at the neuron surface in an adult mouse in vitro model. So we hypothesized here that if the integrin recycling pathway was similarly affected in ERG neurons, we may see decreased levels of integrins at the surface of the neurons. Using primary cultures of the dissociated DRG neurons, we can carry this one step further from what we did in the Drosophila model and directly measure the changes of integrins at the different cellular location by biochemical approaches. This is exactly what we have done by using a cell surface biotinylation assay. Cell surface biotinylation assay isolates all proteins expressed at the surface of the neurons and compare it with the total level of the proteins in the cell. From doing this, we found that the measuring of the total level of ITGB1 showed no difference in all time points, but we found a consistent decrease in the levels of ITGB1 at the surface of the cells by paclitaxel treatment in our Drosophila model at all observed time points. These results indicate that, as implicated in our Drosophila model, paclitaxel perturbed recycling pathways, leading to the reduction of integrins at the surface of DRG sensory neurons in vertebrate CIPN model. So altogether, our results strongly support a model whereby cell surface delivery of integrins is perturbed following paclitaxel treatment, leading to degeneration of sensory afferent terminals in the skin layers. We also show that the neuron-specific overexpression of integrins prior to paclitaxel treatment protected sensory neurons in two different animal models, Drosophila larvae and adult mouse models of CIPN. While it is unlikely that the integrins would be the only receptor that changed following paclitaxel treatment, our results strongly suggest that integrins are key cell surface receptors required for healthy maintenance of nociceptive neurons. So as a step towards translation, we're currently studying the protective capacity of integrins in preclinical adult mouse CIPN models using sensory neuron-specific virus and an integrin activating compound. So following from this work, I'm collaborating with colleagues to develop novel human pain models to study the neuron-substrate interactions. We studied co-culturing human keratinocytes and nociceptive neurons to optimize a 3D skin nerve prep. And using this setup, we ultimately aim to test how physical contact and skin-derived extrinsic factors could influence dysfunction in nociceptive neurons in 3D environment. This is important because our increasing knowledge of the molecular differences in sensory neurons between humans and animal models, and the CIPN and neuropathic pain models that capture human genetics and physiology would add significant advantages. Therefore, it is my hope that my translational strategy using both animals and human models will significantly inform and build foundations for precision pain medicine approaches in the field. A parallel project that I'm currently running together with this neuron-skin interaction project is to understand the immune component in neuron-skin interaction in CIPN. Immune system plays a critical role in neuropathic pain, but how they interplay with neurons and skin cells are just beginning to be explored. Our preliminary results show exciting phenotypes that indicate immune cell transitions throughout distinct neuronal pathological stages, which will form foundation for deciphering neuron-immune cutaneous interaction in CIPN. So that's it for my project. But before I finish my seminar, I would like to make a short advertisement for the research society that I'm actively involved in. It's called the Peripheral Nerve Society, and we are focusing on all pathological conditions involving sensory peripheral neuropathy, and currently have 5 areas of interest, including neuropathic pain, toxic, immunological, hereditary, and diabetic neuropathies. Our current members are mostly from Europe and US, and we hope to increase representation from Asian countries. So if you're interested, please don't hesitate to contact me or contact the society directly to find out about what we can do to help your research. So with that, I'd like to acknowledge my colleagues and mentors for the significant contribution to this project. And thank you so much for your attention. 감사합니다.